Hello everyone. The title of research paper is Evaluation of Full Length versus V4 region 16S rRNA sequencing for phylogenetic analysis of mouse intestinal microbiota after a dietary intervention. Authors Sid K3, Yaya Anwar, Lisa Hoving and all. Presented by Srushti Vingakar. Here is the brief introduction about research paper. Study aim. Study aim is to evaluate if sequencing of full-length 16S rRNA gene using smart sequencing provided different results compared to sequencing only the V4 region using Illumina MI sequencing. Next is about 16S rRNA sequencing. To evaluate the success of intervention, researchers commonly use massive parallel sequencing of the 16S rRNA gene, specifically targeting one of its variable region. The next is next generation sequencing. Next generation sequencing technology like Illumina's MI sequencing provide high throughput sequencing but generate relatively short reads. Then next is third generation sequencing technology. Third generation sequencing technology such as specific biosciences single molecular real-time sequencing, short form is SMRT. Sequencing offers solutions by sequencing longer amplification which can overcome some limitations and short read sequencing. Move to the problem. Sequencing the 16S gene done by feeder diet without and with inulin and from sesame sample by generate two data sets. First one is the PacBio Smart Sequencing. PacBio Smart Sequencing providing longer reads length, smart sequencing outcomes, some limitations of short read technologies. Second will be the Illumina sequencing. Illumina MI sequencing provide high throughput sequencing but generate relatively short reads. Then move to the theory, theory, the theory of sequencing technologies. 16S rRNA gene sequencing. 16S rRNA gene sequencing is to identify and classify bacteria based on conserved and variable regions. There are some limitations. Short read sequencing, while high throughput struggle with resolution closely related bacterial species due to the short read length and limitation is reference databases. Then second is third generation sequencing, which is packed biosmart sequencing. PacBio Smart Sequencing provide long read length. Smart sequencing overcome some limitations of short read technology. Here is the mice were feed a diet without and with inulin. The next is methods. Sesame sample is collect from the mice. Mice is fed with diet and without inulin. Then DNA isolation is done and seen the 16S rRNA by PCR amplification. Then next is the result. First is different amplification length influence observed bacterial abundance. The full length 16S rRNA gene and VPO region capture different subsets of microbial communities affecting the observed relative abundances and certain taxa, as some taxa are better detected by longer sequences. Second is the PCR amplification. The PCR amplification differ between amplicon length, longer amplification like full length 16S rRNA are less prone to PCR basis and amplification artifacts compared to short region, which can lead to difference in observed bacterial composition and abundance. Third is longer amplification length provide different taxonomic resolution. Full length of 16S rRNA include all variable regions, offer a more detailed view of microbial diversity than the shorter VPOR region, leading to the more accurate detection of additional taxa and relative abundance. Fourth is full length sequencing revaluation, unique taxa not detected by VPOR. The full length sequencing kept might miss providing a broad and more unique or rare taxa that short amplification like VPOR comprehensive view of microbial diversity move to the discuss discussion. The first is the address primer and extraction variability. Standardized primer choice and extraction method to minimize available and ensure consistency of sequencing results. Second is to improve the taxa assignment, develop better reference database and analytical tools to assign previously assigned reads and fully utilize the advantages of full length sequencing. The third is validate findings. Conduct further studies to validate the observed differences and bacterial compositions and the impact of sequencing length on taxonomic and diversity analysis. Fourth is optimize sequencing protocols. Evaluate and refine sequencing